Is okay, so it's the correct mode, right? All right, so first we will talk about the basics of genetic testing and how this can be hopefully made a little bit easier for you, because I understand this is not something that is routinely covered in medical school. Okay. So this is a typical genetic report that most labs would return to. And typically after reading the first paragraph and we go on to the second paragraph, it becomes very difficult to understand. So the purpose of this talk is to try and help you to understand a little bit about some of the words and what it actually means, and hopefully you can appreciate it better. Okay. So the outline of my talk will cover the essentials of molecular biology and what you really need to know as a pediatrician and a nephrologist, the basic interpretation of a genetic report, and then appreciate the process of how we determine whether a genetic variant is considered disease causing or not. So first, I will talk about the essentials of molecular biology. So as we all know, proteins are the building blocks of life, right? So from the cells, all the organisms are made out of cells, which are essentially proteins, right? So genes direct the manufacture of proteins. So we have genes like, for example, podosin, and that will produce protein, the podosin protein in the cells, and then these ultimately are grouped together into cells, and then it becomes a living organism. So genes is really about the DNA. So as we all know, in DNA, we have the double helix, right? So it's essentially two columns of nucleotides put together in a helix. So pretty much like the DNA bridge that we have in Singapore, right? So you can see, you can appreciate the helix here. But when we put this out together, it's actually a string of nucleotides, right? So there are 4 types of nucleotides here. In the DNA, the nucleotides will be adenine, which is A, thymine, which is T, and then we have C, which is cytosine, and then G, which is guanine. So it always binds as A to T and C to G, right? So in RNA, it's a little bit different. It will be A to U, because the thymine becomes a uracil, and the C remains binding with guanine, right? So, so to put it in context, we have one strand of DNA, which is here, right? You can see is one strand here. It binds with a second strand, and together in between they have bonds. And then this entire 2 strands are then curved or rotated a little bit to form a helix. Similarly in RNA, it will also be 2 strands as well. It's just that the T is replaced by a U. Okay. So just to recap, in genes, we have nucleotides. Then the nucleotides will then code for amino acids, and the amino acids will then form proteins, which can form the organism. So if there is a change in the nucleotide, right, we used to call this a mutation, then it will result in a change in the amino acid. And if this is an important amino acid, it will then affect the function of the protein, and the organism will then manifest this as a disease. So how do we tell whether the nucleotide is changed, right? So we actually have a reference sequence, right? So the human genome has been sequenced about 1 or 2 decades ago. And so we have a reference. So if there is a difference from that reference, then that is considered a genetic variant. So essentially a genetic variant is when we have a nucleotide sequence that is changed or different from the reference human sequence that we have. So that is a genetic variant. And this genetic variant may or may not be disease causing. Okay, and so a genetic change actually occurs in many ways. It can occur at a chromosomal level, where you have things like chromosomal 21, or even microdeletions, which is at the chromosomal level. Then the DNA change can also be at the DNA level, where sorry, so in a chromosomal level, when the changes will be a deletion of the chromosome, an insertion, an inversion, or a translocation of the chromosomes. Whereas in the DNA, in the nucleotides, the changes can be in the form of a substitution, a deletion, duplication, insertion, or inversion. Then on top of that, there may be copy number variation. Copy number variation means there could be this particular strand of sequence that is just repeated more times. It means it's maybe instead of occurring once, it occurs 2 or 3 times, or it could be occurring less, meaning instead of occurring 1 time, it didn't occur at all. So that's a big deletion, right? So I think one of the most important concepts that we need to understand is that not all genetic variants cause disease. So we have billions of nucleotides within our DNA. Almost all of us would have, thousands and thousands of genetic variants in our genome, and yet we do not manifest diseases because most of these variants are benign and because they do not affect the function of the protein. Right, so it is important to know that not all genetic variants identified are pathogenic. Okay, and the reason is because these changes of the nucleotide may or may not affect the amino acids, right? It may not result in a change in amino acids. And even if it does result in a change in amino acid, it may occur in the part of the protein that is not so important. And therefore the function of the protein is not affected. And therefore the organism remains healthy. Okay. It is also important to know that the word polymorphism and mutation are now outdated terminology. Most people now do not use these two terms. In the past, polymorphism used to be defined as any genetic variants that occur with an allele frequency of more than 1% in the healthy population, whereas a mutation is considered a mutation if it has an allele frequency of less than 1% in the healthy population. But now with next generation sequencing, we know that this is not true. An allele frequency, a genetic variant that occurs at an allele frequency of less than 1% may not necessarily be a pathogenic variant. Okay, so the modern term now is genetic variant, and this genetic variant can then be classified into one of these 5 categories, right? Pathogenic, likely pathogenic, uncertain significance, likely benign, or benign. Okay, so number 3 is actually what we call the VUS, right? Which is variant of unknown significance, something that you probably will hear very frequently about. Okay. So let's just understand a little bit about genetic variants first, right? So in order to do this, we must first understand the basic structure of a gene. So imagine all these blue circles here are actually nucleotides, right, within the DNA strand. So a certain section of the gene would be exons, right, determined by the green ones. And so for a particular protein, there may be several exons. And in between the exons, there will be nucleotides that are actually introns. So these introns generally do not affect protein at all. Okay, so they are introns interspersed by exons interspersed by introns, and then there's a head and a tail. So that's the N terminal, the 5' terminal as well as the 3' terminal. Okay, so then at the beginning of each gene, there will be an untranslated region. Again, is what we call a 5' UTR. And then at the other end, at the tail, there will be a 3' UTR. So these orange sections are pretty much like the red sections. They are non-coding for a protein. Only the green sections, which are the exons, are coding for a protein. And then of course there's a promoter region, and that's exactly where the protein is started to mimic. That's the beginning of the amino acids. Okay, so when we talk about a podosine gene, that's how a structure of a typical gene looks like, right? So you have green with red and then the 2 orange at the 2 ends of it. Similarly, the collagen 4A5 gene is a bigger gene. So podosine is a very small gene, only 8 exons, whereas in the collagen 4A5 gene, it's a very big gene with 58 exons. Okay. So the most important part of the gene is really the exons, right? Because it is these exons that eventually will code for the sequence of the amino acids in the protein. So this is what we call the coding regions. Okay, so the less important parts of the genes would be the red sections and the orange sections because they are non-coding, and that's because they do not affect the sequence of the amino acids, and they generally are thought to be less likely to cause diseases. However, there are still genetic variants within these non-coding regions that can still alter protein function. And how does it do it? Is because it can alter the level of gene expression all through the process of splicing. Okay, so intronic genetic variants rarely can still be disease causing. Okay, next we need to understand the central dogma of molecular biology. So this basically means that we have DNA in our cells, or the nucleus of our cells. They actually undergo transcription to form RNA. This RNA then undergo translation to form protein in the cytoplasm. Okay, so when we talk about a genomic DNA, which is really the DNA within the nucleus of the cells, okay, so as I said, you have the orange, the green, and the red sections. When we form a coding DNA basically means that there is only the exons string together, and then that will then form the RNA, which is the transcript, right? So this entire process is called transcription. Now, it's important to identify the word genomic DNA versus coding DNA, because that's how you will eventually interpret the nomenclature of the variants later. Okay, so genomic DNA is G, coding DNA is C, cDNA versus gDNA, right? Then from the RNA, it is then coded right into amino acids. So this is what we call a codon, right? So 3 nucleotides will then form a code for 1 amino acid, and there's a particular code that we need to know, right? So with that, then the amino acids are formed, and that will be strung together to form a protein. Okay. If we have one genomic DNA, one strand, it can lead to different types of RNA transcripts because of the process called splicing. So RNA transcript 1, right? So because of splicing and the way the transcription is being done, we can form RNA transcript 1, where you can see the exons 1, 2, 3, 4, 5 are put together. And then these transcripts are then given a name, right? So as you can see here is NM something, something, something. Okay, so this is a particular transcript. Now, if you, sometimes because of splicing, there may be another form of RNA that will result from this genomic DNA. As you can see here, as a result of splicing, exon 3 and exon 5 are removed. And this forms another transcript, a different transcript on transcript 1, and it can be given a separate name and number that is different from this particular transcript. Okay. So similarly, it can have a third transcript here where you can see exon 6 and exon 5 are dropped, and it is given a different number. So when you see genetic reports nowadays, the transcript number may be included, and you recognize this by this NM number. Okay, and the reason, and it's important to know because the numbering may change if you use a different transcript. The numbering of the nucleotides and the amino acids may change if you use a different transcript number. Okay, all right, so let's move on. Okay, so now let's just talk a little bit about translation here, right? So the conversion from RNA to amino acids. So as I said here, the RNA transcript doesn't have any introns. It's a RNA nucleotides, and it's all divided into 3. And each group of 3 is called, will be a codon that code for 1 amino acid. And this is according to this particular algorithm or script that is just the way we are made, right? The way we are made is such that the RNA nucleotide UUU will code for phenylalanine, I think, whereas say a CCU RNA put together, nucleotide RNA put together will form a proline amino acid. So this is a particular code, right? That the way, and that's the way living organisms are made. Okay, so we have the codes here, and this is the different amino acids that it codes for. Okay. So when we put in the string, right, the different thing, it is labeled according to either a 3-letter code or a 1-letter code, right? So you can see that each amino acid is coded by a code. Okay. And so when you see the protein sequence, it is determined by these letters here, right? So M, R, S, Y, Z, L, K. So M stands for methionine, which is always the first amino acid of every protein. Then R would stand for arginine, and then S would stand for serine, for example. So that's how it is being labeled. Okay. So as you can see here, we have a starting point as well. So the genomic DNA, the starting point would be somewhere here, and in the coding DNA, it will be an ATG, right? And in the RNA, it will become an AUG because the T becomes a U. So this is the start of the first amino acid in the protein. So this is what we call a start codon. It will code for methionine, which is always the first amino acid in a protein. Okay. And so the numbering will start from the starting point of the genomic DNA. So as you can see here, the red circle will determine the starting point of how you number the nucleotide or the amino acids. Okay, so this numbering is important because the starting point of how you number the genomic DNA is different from how you number for a coding DNA. Okay. So this is the start codon here. So every protein starts as AUG, start codon, and it will end as with a stop codon. So stop codon will be UAA, UAG, or UGN. So that will determine the end of translation or the end of the amino acid chain. Okay, so as you can see here, this is the start codon, and then these are the stop codons. Okay. All right, so now let's move on to the interpretation of a genetic report. So this is a typical way, a typical a genetic variant, right? So G.13C2A, sometimes it's written as G.C13A, which is the same thing, right? G.C13A basically means the C nucleotide at position 13 is changed to A. Okay, so remember, if it's a G in front, it determines a genomic DNA, not coding DNA. It's important to know that because when you count number 13 is here, it's based on the start nucleotide starting from here. Okay, so the starting point is different. And in this particular case, this C at position 13 is changed to A, right? So yeah, so as I said, it can be changed. It can also be written as C G.C13A means the C at position 13 is changed to A. And so this is called a missense mutation, which is basically a substitution of a single nucleotide, right, from one to the other. Now, genetic variant is usually written as this way, a C.8C2A. So in this case, it's a C, right? It's not a G. So this is a coding DNA, right? So coding DNA has no introns. It only has exons, right? So it will start with a ATG, right? And in this case, because it's number 8, means we're looking at the 8th nucleotide, taking the position 1 as the ATG here, okay? And then C to A means at position 8, nucleotide number 8, the C is converted to an A, right? So it can also be written as C.C.A.A. Okay, and this is the same missense mutation as G.C13A, okay? Because there is no new intron here. There is no intron. This is the first exon, right? Whereas if this is the second or third or fourth exons, then there's in between introns, then the numbering would be different, right? It could be G.C 135 A could actually be C dot C25A, right? So the C will be shorter because it doesn't have introns. Okay, so bringing this to the protein level, right, the coding DNA would then be transcribed into the RNA. So similarly, the RNA would be changed at the same position. So in this particular case, you can see that the position 8 here, right, is changed to an A, right? And if you use the coding here, by changing this C to an A, it actually results in a change from alanine to aspartate. Okay, so alanine to aspartate. So in this case, that's how a genetic variant can also be written. So it's a P dot alanine to aspartate, right? So P here stands for protein. So this is about protein. It's not about coding DNA. So in a protein, at amino acid number 3, the alanine was the original or the correct amino acid, but that has been changed to an aspartate. So then the genetic variant is written as P dot ALA3ASP. Okay. Or it can also be written as a one letter code, right? So in this case, alanine is denoted as A and aspartate as D. So therefore, this genetic variant can also be written as P dot A3D. Okay, they mean the same thing. So just to recap, G dot C13A is the same as C dot C8A because there's some introns being removed. Then that would be equal to a P dot A3D, means the third amino acid in the protein is changed from an A to a D, right, which is alanine to aspartate. So this way of naming the genetic variants, right? There are different ways and they actually mean the same thing. Okay. Okay. In this case, it is a non-synonymous missense mutation, which means that non-synonymous means that there is actually a change in the amino acids, a change from alanine to aspartate. So it's called a non-synonymous, right? It's not the same. But some amino acids may not change. So give you an example. If I have this particular variant that is a C14G, for example, so instead of 13 is a 14, and that results in a C.CG9G. Okay, so in the RNA here, you can see that at the 9th position, the C is changed to a G, right? So instead of a GCC is now a GCG. And when we look at the code here, actually, whether it's GCC or GCG, both of these codes will code for alanine. So even though the nucleotide did not, even though the nucleotide leads to a change, right? There's a change in the nucleotide, there is no change in the amino acid coded. So in terms of the protein wise, it remains the same. Okay, so as you can see here, this one would then be a, because the A remains as an A. So that tells you that this is a synonymous missense mutation. Synonymous because the amino acid remains the same. Okay, so this is a synonymous missense mutation, whereas this is a non-synonymous missense mutation. Now, so if you look at these, right, if you have a P dot alanine 3 to aspartate or a P dot alanine 3 to valine, then this is considered non-synonymous. Okay, whereas a synonymous mutation is alanine, it remains as alanine, so it's a synonymous. Okay, the other thing is that's interesting is that if it's a alanine to aspartate, it is actually charged, right? Aspartate is charged. Is a charged amino acid, whereas alanine is a hydrophobic. So it is likely that because there's a change in the charge of the amino acids, that it will change the properties of the protein and therefore more likely to result in a change in the function of the protein. Whereas here, the ALA to a VAL is hydrophobic to hydrophobic amino acid. So it is less likely to cause a change in the amino acid, in the protein function. But this is really by pure prediction or computer modeling or algorithms. But this is one of the things that people do in order to determine whether a genetic variant is likely to be pathogenic or not. And this is seen by what you see as in silico algorithms, right? So you may hear of things like polyphen, that are essentially just computer modeling that take into account all these little changes in the amino acids in order to give to a score to decide whether the variant is pathogenic or not, right? So if there's a change in the charge of the amino acid is deemed to be more pathogenic than if there is no change in the charge or if there's no change in the amino acid. Okay, next I will talk a little bit about deletions, insertion, duplication, inversion. Okay, so genetic variants can also occur as a deletion, right? So it will be denoted as this, right? C.4_5DEL, right? So what this means is that at position 4 to 5, where it's originally G to A, this 4 to 5 amino acids are now gone, is deleted. Okay, so then that will result in a RNA that has again missing nucleotide 4 and 5, and then that will then lead to a different coding, right, of the amino acids. So the amino acids will be changed, and therefore it's likely that the protein function will be changed. So it's important to recognize that this is a frame shift mutation of genetic variant. So what this means is, because as I said, right, the nucleotides are coded in groups of 3 because 3 nucleotides code for 1 amino acid. So if I just give you this, for example, these letters that are actually the cat ran out, the cat ate the rat and ran out. So you string them together like that, right? But actually it made sense. Now, if we have a deletion that involves 3 nucleotides or even 6 nucleotides, because it's in multiples of 3, the sentence still made sense, right? It can be the cat ate rat and ran out, right? So there is no frame shift. The sentence more or less made sense. The protein function may not be so severely affected because it is an in-frame deletion. Now, if the deletion occurs in multiples that is not of 3, for example, two nucleotide deletion or 4 nucleotide deletion or 8 nucleotide deletion, right? So not the multiples of 3, then it basically would lead to a complete change in terms of how the nucleotides are coded into the codons. So this is what we call a frame shift deletion, and the sentence basically becomes nonsensical. So this is what we call a frame shift deletion. Okay. So it's usually considered very pathogenic. So similarly, the same thing happens for insertion, right? So in this case, INS stands for insertion, which means that, in nucleotide position 4 and 5, there is a repeat, right? So instead of 1, 2, 3, 4, 5, it becomes 1, 2, 3, 4, 5, 4, 5 again. Okay, so there's a repeat of 2 nucleotides. And in this case, it will also result in a frame shift because it's not a multiple of 3. Okay, so it's similarly just a cat ate the rat and ran out. You put in a insertion that is in multiples of 3, the sentence still makes sense. The protein function will be minimally affected because it's an in-frame insertion. Whereas if we insert, say, just a 2 nucleotide or a 4 nucleotide, outside of a multiples of 3, then the sentence doesn't make sense. The protein is a very nonsensical protein. So this is a frame shift insertion, and is usually a very disease-causing variant. Okay, so frame shift mutations are likely to be very pathogenic, especially if it occurs at the 5' end. 5' end is actually the head, the beginning of the protein. right? The beginning of the protein is a 5 prime, the end of a protein is called a 3 prime. So if the frameshift mutations occur at the beginning of the protein, then it's likely that the entire protein or almost the entire protein is going to be affected, right? Whereas if the frameshift mutation occurs towards the end of the protein near the 5 prime end or the C terminal end, then it's likely that the effect on the protein is less because it's only at the tail end that it is nonsensical, right? Okay, other kinds of genetic variants would be a duplication, right? So again, is 45 at position 45 to 42 to 45, that's a duplicate. Okay, so in this case, we can see the ACTG is duplicated. Okay, or it could be a inversion, right? So 42 at nucleotide position 42 to 45, there's an inversion, means instead of ACGT, it became TGCA. Okay, so indel, okay, this is a little bit more complicated, but indel basically is actually insertion deletion, and so it can occur within the same at the same time, right? So in this particular case, there is an insertion as well as a deletion occurring at the same time. So in this case, for example, if you have this sequence and if you have this indel, then it means that instead of the ACGT is deleted, and at the same time A and T is inserted. Okay, so this is what indel means. Okay, so this is the final sequence here. Okay, so a nonsense mutation or nonsense mutation basically means that we have a missense mutation that results in a premature stop codon, or a frameshift mutation that results in a premature stop codon. So the protein is prematurely stopped because of the mutation, it somehow happened to code for a stop codon. Okay. So like, for example, in this case, right? So in RNA position 30, let's say that it was UGG, right? And let's say the G is changed to an A, then instead of coding for tryptophan, UGA actually codes for a stop codon. So then the amino acid will stop right here, and the entire protein will be truncated. Right, as you can see here, instead of 10, 11, 12, right? After 10 amino acid, everything is stopped already. So this is what we call a truncating mutation or a nonsense mutation. And usually these are quite pathogenic. Again, especially if these occur at the earlier part of the protein. So the head of the protein, beginning of the protein, may be less likely to be pathogenic if it occurs at the tail end of the protein. Okay, so with this, I hope this sentence becomes a little bit more clearer, right? So when you read at least the first statement here, right, a variant was identified in this, you can see KCNJ1 is actually the gene, and M followed by the number is actually the RNA transcript, and this is actually the variant, right, coded, given as cDNA coding, as well as a protein coding. So you can tell that at nucleotide position 658, the nucleotide was changed from a C to an A, and at amino acid position 220, the LEU was changed to PHE. Okay, so if you want to have more information, you can actually look up this website here that will actually tell you the different nomenclature that people use to understand the genetic variants. Okay. All right, now let's just move on to the third part, which is probably what a lot of you are interested in, is to understand the process of how we determine whether a genetic variant is disease causing. So in this case, you have a genetic variant. As I said, not all genetic variants cause disease. So how do we decide which one can cause disease? So again, as I said, it's divided into the 5 categories here. So a variant is considered benign if the likelihood of it causing disease is less than 1%. It's considered likely benign if the likelihood of it causing disease is 1 to 10%. So similarly, it is likely pathogenic or pathogenic if the likelihood of causing disease is more than 10%. So in between, at 10 to 90%, you have variants that lie in between here. There's really no man's land, right? And this is what we call variant of unknown significance. Essentially, these are considered inconclusive and have no genetic, or rather no clinical significance. In other words, it is not correct to use variants of unknown significance to direct clinical treatment, right? A variant of unknown significance is not considered a pathogenic variant and should not be used to direct management of the patients. So how do we classify these genetic variants? It's really using the classifications determined by this particular paper here. I mean, you all can read it out, but it may be a little bit confusing for you, right? So this is the most important table in that paper, where it tells you that when we classify variants, we use criteria. Those criteria are divided into benign and pathogenic based on various evidence here, right? So it can be population, computing, functional, etc. So I'm just going to go through these with you one by one to just help you understand, or at least appreciate what this is all about, okay? So to determine whether a genetic variant is disease causing, we use different groups of criteria. And these groups would be the nature of the variant. So as I said, whether it's a missense, whether it's frame shift, whether it is truncating, etc., etc. Okay. Then we talk about family segregation, whether this variant occurs in the family members that are affected and not in those who are not. Then it also has criteria that involves whether this variant occurs in the healthy population or whether it has been determined by scientists in cells or in animals and deemed to be pathogenic. So then also the allele frequency in the healthy population. So there are that large databases that involves healthy population where they then look at how many times does this variant occur when we sequence a healthy population. Next, we talk about in silico tests. So these are essentially computing modeling tests to try and at least theoretically predict whether the genetic variant is likely to be pathogenic or not. Okay, and then finally it will be research, right? So people, whether they have looked into animal or cell models to see whether the genetic variant affects the function of the cells or the animals. Okay, so okay, nature of sequence of variation. So as I said before, right, the frame shift deletions and the nonsense mutations. So nonsense means the truncating one, those that lead to a premature stop codon, or those that involves multiple exon deletion. So many exons are deleted, or canonical splice sites. So canonical splice sites is actually variants that occur right at the junction between the intron as well as the exon. So it's at the junction between the intron and the exon. So where the exon is counted as number 1, where at the end of the intron is -1, right? So that around that junction when they have and the genetic variant occurs around that junction, it is predicted that it will then affect the splicing of the RNA, and therefore it will result in a significant change in the protein. Okay, so variants that occur in the canonical splice sites are considered likely to be pathogenic. Whereas the missense mutation generally are less likely to be pathogenic. However, the non-synonymous ones, means those that lead to amino acid change, can still be pathogenic depending on the criteria of the missense mutation. Whereas the synonymous missense mutation or the intronic variants are less likely to be pathogenic, but the likelihood is still not 0. Okay. Now, all of us have 2 copies of most genes, right? So 1 copy of the gene from daddy, 1 copy of the gene from mommy, except for the X and the Y chromosomes, right? So what can cause disease? So if it's an autosomal dominant gene, then just 1 abnormal copy is enough to cause disease. So if it's an autosomal dominant gene, a heterozygous individual can be considered to be to carry a genetic variant that is disease causing. In autosomal recessive genes, you would require 2 abnormal copies. So a heterozygous variant, variant does not necessarily mean that the individual will have the disease if this is an autosomal recessive condition. So for autosomal recessive conditions, the individual must have 2 abnormal copies to be deemed as having a disease-causing variant. So these 2 abnormal copies can be homozygous or it can be a compound heterozygous. In a sex-linked recessive condition, then you would need 1 abnormal copy in the male because it occurs in the X chromosome. And these males would then be called hemizygous, right? Because they only have 1 X chromosome, and that single abnormal gene copy in the X chromosome is enough to cause disease. So these are hemizygous males. Whereas in females, they will have to be homozygous because they will need to carry both 2 abnormal gene copies in order to cause disease. Now, if it's an X-linked dominant condition, then 1 abnormal gene copy is considered enough to cause disease. So either the hemizygous males or the heterozygous females can have disease. Okay, so one thing here that I have missed out is under here in autosomal recessive cases, an individual can have homozygous variants, meaning the same variant occurring on both gene copies, or it could be compound heterozygous. So the individual carries 2 pathogenic variants within the same gene. Now, in this case, this is called compound heterozygous. Then you will need to sequence the family, particularly the parents, both parents, to determine whether these 2 pathogenic alleles occur on the same allele or on different alleles, right? So that means whether it occurs in cis or whether it occurs in trans. So if it occurs in trans, means each parent carries 1 pathogenic allele, and therefore the patient is likely to have an abnormal pathogenic variant on each allele. Whereas if they are in cis, then both are occurring in the same allele and therefore are less likely to cause disease. Okay, let's move on. So how to tell a variant is significant? So in this particular case, right, so family, you can see the gray are the ones who are affected. And so if you have a variant that occurs only in the affected individual in the family, but in the unaffected individuals in the family, you sequence them, you found that they do not carry the variant, then this is clear that the variant segregated with disease in the family. So those who have disease carries the pathogenic variant, whereas those that do not have the disease does not carry the pathogenic variant. So this is called good family segregation, and therefore the variant is more likely to be pathogenic. So in this particular case, okay, if this particular variant only occurs in these 3 individuals here, right? And then if you sequence and you find that these 2 individuals do not carry the variant, then this variant does not segregate well with disease in the family, because you don't see that, only the affected carries the variant, right? So in this case, this variant is less likely to be pathogenic. Okay, now we talk about databases, literature, and the minor allele frequencies in the healthy population. So there are actually disease databases. So you may have heard of ClinVar, OMIM, human gene mutation databases. So these are databases where people contribute to if they find pathogenic variants. So these are, for example, you sequence, you have a sequencing company, and then they have a particular patient that has this particular case, and then they found a novel mutation, right? Novel genetic variant. And through the various criteria, they determine that this novel genetic variant is likely to be pathogenic. So all these sequencing companies will then deposit all these identified genetic variants into databases like these, so that other people in the world can then look at these databases. And if they happen to have a similar patient with a similar genetic variant, then they are more likely, or at least more confident, to say, hey, this variant I'm seeing in my patient now is likely to be pathogenic, because somewhere in the world out there, there's another individual with the same genetic variant with the same condition as my patient, right? So this is how doctors and scientists contribute and work together to help in the variant curation. Okay, then of course you can look at PubMed, where there may be case reports or studies, to describe the different genetic variants and the phenotype of the patients. Okay, so the other thing we look at is healthy populations, right? So there actually have been large studies where we look at healthy people across different populations in the world. And if we have if we sequence 50 healthy populations, that's actually equal to 100 alleles, because one person has 2 alleles. Okay, and if we find that genetic variant in 3 out of these 100 alleles, then the allele frequency is 3%. Okay, so just give you an idea. So we have 100 people here, and there are 3 people in this 50 people that carries this particular genetic variant, right? And these people are assumed to be healthy, assumed not to have the disease that you are interested in, right? So in general, we believe that a pathogenic variant should have a very, very low allele frequency in the healthy population, because if this is truly pathogenic, it should have caused disease in that person to make him not eligible to take part in this particular study. Okay, so you actually can look up databases. So actually the common ones that people use nowadays is the NOMAD database. Okay, so is this one here, yeah? This one, this EXACT is very outdated already. It's actually the NOMAD, which actually incorporates data from the EXACT. Okay, so the NOMAD databases actually also have different populations across the world, right? So what we are interested in, or what you will be interested in, would be the East Asians, right? Because Vietnamese would have qualified or would be quite categorized as East Asians. Okay, so we look up these databases to see whether this particular variant you're interested in, whether it has been identified in this database or not. All right. So as you can see here in this report here, right, this variant is present in 0.224% of the South Asian population as reported by this database. So you can see that this database, when you look at the South Asian population, 0.224% of the healthy population carries this genetic variant. Okay. Okay, next we talk about in silico tests. So in silico test is actually many things. You may have heard of things like polyphen, CAT, SIFT, mutation taster. So these are actually just computational modeling or algorithms where they look at things like whether the mutation is synonymous, whether it causes a change in the charge of the amino acids. Then for GERP, they also look at whether the variants occur are evolutionarily conserved, okay, which I will explain to you later on. So by using this computer modeling, right, they basically use the location of the variant in the context of the protein sequence, the biochemical consequence, and also the evolutionary conservation to give a certain score, and then at least theoretically decide whether this variant is likely to be pathogenic or not. Okay, so if we have this particular protein sequence here, so right, this is a protein sequence denoted by the amino acid codes, and this is at position 60, position 70, position 80. Then we looked at the sequence in the different organisms here, right? Different organisms. If you look at these particular positions in green, you can see that they are identical across all different species. this suggests that this part of the protein is likely to be very, very important for the function of this protein, because across all living organisms, right, or many living organisms, is conserved. So it's likely to be an important part of the protein. Whereas if you look here, you can see that this part of the protein is not evolutionarily conserved, right? So therefore, this is likely to be a less important part of the protein. So variants that occur in the red part are deemed to be less likely to be pathogenic, whereas variants that occur in the green parts are considered to be more likely to be pathogenic. Okay. Okay, then of course there may be research because there may be scientists that would then make the mouse have this particular variant that you're interested in, or put this particular variant into cells and see whether the behavior of the cells or of the human or of the animals actually change or not. And with that, you can also then help to decide whether the variant is likely to be disease causing. Okay, so essentially, oh dear, essentially, if you have a variant, right, particular variant, you want to see whether it's pathogenic, we actually, oh dear, what did I do? You actually can assign criteria, okay, such as these, to see whether they are likely to be pathogenic or not. Okay, so according to this particular standard paper that I've talked to you about, right? So this case, you can see I've grouped them together. When you look at the nature of the sequence, the evolutionary conservation and computer modeling, the allele frequency, the protein functional domains, right? So which part of the protein is likely to be functional and which part is not. So all these will help you assign specific criteria. Then there's another group where you look at whether it has been reported in case control studies in the literature, whether the scientists have looked at it in cells or in animals. We looked up the healthy population databases or the literature to see whether it has ever happened in healthy or in the diseased individuals before. Okay, so in general, the red part here, right, most sequencing companies will be able to do it for you because they would have certain standard algorithms of doing it. Then the yellow part here, in general, most companies would be able to do it for you because they would have also looked at these studies in general, okay, although not everybody does it the same way, right? But you as a clinician at the bedside can also look at these particular criteria like the phenotype specificity, whether the phenotype of your patient fits into the disease of interest, whether the variants segregate with the family, meaning whether the disease family carries the variant, whereas the non-disease family members do not, and also whether it co-occur with other variants, either in cis or in trans. So later I will illustrate these principles further with some of my examples cases, okay? All right. So you suspect a patient has a particular syndrome, you send the patient for a genetic test. Well, it can come back as some genetic variants found. You decide whether they are disease causing based on these criteria that I have mentioned, right? Or it could be no pathogenic variants found. So the question is it really negative? So you need to understand that a negative genetic test report does not mean that your patient does not have the genetic condition. Okay, in other words, there can be false negative. Okay, and why could there be false negative? Because there are actually genetic variants that can be missed in our usual genetic testing. For example, if the patient has very large deletions, so like deletions that involves many exons or almost the entire gene, or if there's large duplications, these may not be picked up by the conventional next generation sequencing method, unless there have been efforts to change the bioinformatics of the next generation sequencing in order to pick up some copy number variation. But even then, there will still be limitations. So if your patient has, say, a very large deletion, or if you're doing a whole exome sequencing, but your patient has a very important intronic genetic variant, it will also not be picked up. Okay, so in those situations, you may then consider other methods of genetic testing like a long-range PCR, MLPA, duplication deletion analysis, microarray, or whole genome sequencing, right? So later I will touch a little bit about whole genome and whole exome sequencing, but basically in whole genome sequencing, you sequence the entire genomic DNA, which means you would sequence the coding as well as the non-coding parts of the DNA. In whole exome sequencing, we only sequence the coding parts of the DNA, which is literally only the exons, right? So if your patient has an intronic variant or a splicing variant that is outside of the canonical splice sites, it will not be picked up by the conventional whole exome sequencing or most gene panel sequencing, unless you do a whole genome sequencing or do special types of genetic testing. Okay, so with this, I would like to end my lecture here. Are there any questions? I have another section. Are there any questions? Or we can leave the questions to the end of the lecture. Oops. Okay. Can you see my slides? Yes. Okay. So so the next part of my talk will be about genetic testing in the context of glomerular disease. Maybe sorry, the top type is a little bit different. It should be actually genetic glomerulopathy lah. Okay. All right. So as you know, primary nephrotic syndrome actually involves, usually at least in pediatrics, is the initial treatment with steroids, 4 to 6 weeks. If they're responsive, that's okay. If they're not responsive, which occurs in about 15% of children, then they are considered to have early onset SRNS. Okay, however, among the responsive groups, there may be some patients that actually go on to develop steroid-resistant relapses, and these patients are deemed to have late onset SRNS. So SRNS is an important condition because it can account for about 10% of early onset CKD. So in general, SRNS is divided into two groups, the genetic group as well as the immune-mediated group. The immune-mediated group is considered important because 30% of this group actually have post-transplant FSGS recurrence. Among the genetic group, how common are genetic variants or pathogenic variants? So really in a pediatric SRNS cohort, most studies would show that about 20 to 30% of such patients actually have a genetic etiology. And this is by far patients who have initial onset steroid-resistant nephrotic syndrome. Those who have late onset steroid-resistant nephrotic syndrome are generally thought that they do not have genetic etiology. You know, if your patient has a family history or comes from a consanguineous family, then the likelihood increases, okay, to half or two-thirds. In adults, it's roughly about 10%. Okay, so in adults with primary FSGS, roughly about 10% of them have a genetic etiology. Whereas, hold on, yeah. Whereas if in adults that have family history, then the likelihood of a genetic etiology is even higher. Okay, so however, I do note that there's actually a large heterogeneity of the studies. So really, when we look at the literature, it is not really 20 to 30%. It really can range between 10 to 40%. And it really depends on the inclusion criteria of the study, the definition and how the phenotypes are evaluated, the types of genes tested, how the genetic test is done, and more importantly, how the pathogenicity is determined, right? So in the earlier studies, they may not use the ACMG criteria to determine the pathogenicity of the variants, okay? So as I said, the initial onset sRNS, about 10 to 30% of them have a genetic cause. Whereas in a late onset sRNS, generally they are not considered a genetic cause. Okay, so in the initial onset sRNS, about 30% will recur after transplant, whereas the late onset sRNS, up to 90% may recur after transplant. So this group is nowadays regarded as an immune-mediated group. Okay, so genetic testing is now recommended when once a steroid or at least initial steroid resistance is determined, right? So if the patient first presents with nephrotic syndrome, you treat the patient with 4 weeks of high dose oral prednisolone, and there is no remission after 4 weeks, you should then consider doing genetic testing for your patient. In the KDIGO guidelines, they would suggest that sRNS at children less than 2 years of age, those with syndromic features, or those that have a positive family history, we really should consider genetic testing. So it is important to clinch a genetic diagnosis early in the course of sRNS, because in pediatric nephrology, if the patient is deemed steroid resistant, we would want to give very potent immunosuppression. And these are not only expensive, but can cause many side effects, right? Such as these opportunistic infections, as well as even malignancy later on. And yet most genetic cases do not respond well to immunosuppression. So if you found the genetic case early, you will not have given the patient very potent immunosuppression. And therefore, you not only save costs, but you also save the patient from having to go through immunosuppression and be subjected to very serious side effects. sRNAs cases generally, if they are treated with cyclosporine, if it's a non-genetic case, they are likely to respond well, right? So 21% of a non-genetic case do not respond to cyclosporine. But if the patient has a genetic mutation, it is less likely, much less likely to respond to cyclosporine. Okay, so most genetic cases do not respond well to cyclosporine, but there will still be some cases that will respond to cyclosporine. Okay, so early genetic testing can guide immunosuppressive strategy, right? So most of us would feel like, hey, I'm not trained in genetics. What should I do? And what are the pitfalls? Right, so from the point of a nephrologist here, I'm going to try to help you understand the different nephrotic syndrome genes from a clinician's or a nephrologist's perspective, and how this can impact on your clinical practice. We will talk about which patient is most likely to have a genetic etiology. We talk about how genetic testing can help your patient. Then we talk about what are some of the important considerations for your patients when you want to do genetic testing. So firstly, to understand the nephrotic syndrome genes, right? So the first podocyte gene was found in 1998, and that was using positional cloning. And then 2 years later, the second gene was found, and that's podocin, again using positional cloning. But a few years later, the new technique called next generation sequencing emerged. So in next generation sequencing, it's a little bit different because it allows us to sequence many genes at the same time. Whereas in Sanger sequencing, which is the older form of sequencing method, only one gene can be sequenced one at a time. So next generation sequencing is a lot cheaper and faster and more efficient, right? So because of this more efficient technique, in the last decade or more, we have witnessed an explosion of gene discoveries in nephrotic syndrome and proteinuria diseases, right? So to date, there are more than 50 genes, in fact, nearly 100 genes that have been said to be important in nephrotic syndrome. So in nephrotic syndrome, as we all know, the most important cell is the podocytes, right? So in these genes, if you try to understand it from the molecular point of view, it becomes very difficult to a clinician, because you see the genes can be divided into the actin cytoskeleton protein, the mitochondrial protein, the lysosome protein, the actin regulation protein. It becomes very difficult to be understood by a nephrologist. So how do we approach this list of genes as a nephrologist, right? So I think what I would want to help you understand in this lecture is to help you understand how you can perform a comprehensive clinical evaluation in the patient and the family, so that you have an idea which genes are most likely in the patient. Then you can select the most correct test. Okay. And in doing so, we can then decide, right, based on the presence of extrarenal manifestations, whether we should look at the syndromic genes. Based on the age of onset of disease, we can decide which genes are more likely. Based on the family history, we can decide based on mode of inheritance, which genes are more likely. We can also look at the kidney histology and ethnicity to decide which are the likely genes. Okay, so let me first go through the first one, extrarenal manifestations and looking at the syndromic genes, right? So there are genes that are thought to be syndromic. Syndromic means they are likely to have manifestations outside of the kidney. So commonly we think about the mitochondrial genes as I've listed here. So mitochondrial genes, the patients may have other manifestations like diabetes, hearing loss, cognitive impairment, epilepsy. Yeah, so these are the things that make you suspect that it might be a mitochondrial gene. Lactic acidosis is the other one. Or it could be genes like that cause early onset nephrotic syndrome. So like the Danish-Dresch, Frazier's, Pearson, right? So these genes would have extrarenal manifestations involving the genitalia. So ambiguous genitalia, abnormal hormones because of the strict gonads. In Pearson, the eyes are affected, right? Now in Alport's, the patients may have eye abnormalities or hearing abnormalities, right? And then there may be genes that cause skeletal abnormalities. So as those I've listed here, those where the patients have abnormal bones, right? And then there are a whole host of other stuff where the patients can have diseases of other systems. So in the majority of these, extrarenal manifestations are pretty obvious, but it's really, really quite rare. Most of the time, we as nephrologists will have to look very hard. Okay, so like in this case, I'm giving you the very obvious cases, right? So if you have patients that have cognitive impairment, you would think, and have early onset nephrotic syndrome, you worry about Galloway-Morway syndrome, right? So that's congenital nephrotic syndrome together with neurological abnormalities. Or you may have patients that have very obvious skeletal abnormalities where you then think of these conditions. Or patients with ambiguous genitalia or abnormal skin, or very abnormal hormonal or skin abnormalities. Okay, but most of the time when we as nephrologists, when we see patients, usually they are quite well other than the nephrotic syndrome or the urine problems. So we really have to look very hard for the extrarenal manifestations. So when we say look very hard, means you do a careful neurological and skeletal survey examination. You may want to consider sending them for a formal ophthalmological examination in order to look for eye abnormalities associated with Alport's MYH9, or perform a hearing test because there are some conditions that are associated with neurosensory hearing loss, When we look at the FBC, you may want to look at the mean platelet volume, because in this MYH9 condition, the patients have large platelets. If your patient has diabetes, especially in a family or in the patient itself, or that if there's lactic acidosis, then you do consider mitochondrial genes. Okay, so the other thing you need to remember is genes that are deemed to be syndromic actually can also manifest in patients as an isolated renal condition. So in these genes that I've highlighted here, the patients may only have kidney manifestations and do not have the extra renal manifestations that are classical in these conditions. Okay, so you need to be aware of that. Now, the age of onset. The age of onset in the patient will give you an idea which genes are common, especially if there is no extra renal manifestation. So if the patient manifests in the first 1 to 18 years, then we think of these genes. Sorry, I forgot to put in those that are less than 1 year old. So those that present less than 1 year old, especially if those that present in the first 3 months, then we would look at the nephrin, WT1, PLCE1, LAMP2, right? So those are the common genes. Now, if the patient presents in the adulthood or late adolescence, then the common genes would be the collagen 4 genes, as well as the other autosomal dominant genes, right? Like INF2, TRPC6, ACTN4. So these are all autosomal dominant genes. Whereas in the childhood genes, they are more likely to be autosomal recessive. So many of you have actually took part in this dragon study, and this paper was just very recently accepted for publication in Clinical Genetics. I think Dr. Wang is one of the authors here. So in this first paper, we actually looked at 183 probands from 17 centers and 6 countries. And we actually found 14% of these probands have a genetic variant that is disease causing. What's even more striking is that about a third of these patients with genetic mutation are due to the Alport genes. In other words, Alport genes are actually very common. Alport mutations are very common. So we should routinely screen for them, especially if your patient has unaccounted for nephrotic syndrome. And more importantly, at least in the dragon cohort, 80% of the patients with the collagen 4 gene defects do not have clinical features suggestive of Alports. In other words, there is no documentation that these patients have neurosensory hearing loss, ocular abnormalities, or glomerular basement membrane abnormalities. I think that's because we do not actively look for them, because we don't see the need to, or because of limited resources, but also because many of these patients are pretty young. So this has been shown in many studies across the world that collagen 4 genes are increasingly recognized to be more important, right? Very important in glomerular disease, even in the absence of extrarenal manifestations. Okay. Now, when you talk about glomerular, genetic glomerulopathy, right? The natural history is such that the patients first have microscopic hematuria followed by proteinuria. And because these are asymptomatic, you only detect them if it is an incidental finding. So you happen to do a urine test because the patient has fever, you want to look for urinary tract infection, and you happen to find microscopic hematuria or proteinuria, which on follow-up was found to be persistent. And then that's when we start to suspect there's something else that's going on. So these patients may then ultimately develop into nephrotic syndrome and then go on to have CKD, although there will be some patients that do not have nephrotic syndrome and just go on to have CKD. Right, so it is important to think where is the disease onset, right? Because you know, the patient may have presented to you with nephrotic syndrome now, but actually before that there was actually proteinuria already, but it wasn't detected. So it's important to go back to the history and ask the parents, you know, have there been times when the child was younger and the doctors ever said that there were some abnormalities in the protein before? Okay, the next thing is to determine the family history carefully, right? So this is very important. Most of us are nephrologists. We are not not geneticists. So when we take a family history, we tend to focus on the patient, the siblings, and the parents. We tend to forget the grandparents and the uncles and the aunties, which are actually the parents' siblings. So it is important to draw a 3-generation pedigree, focusing on also the extended family. And more importantly, you should not ask only the kidney diseases. That means you ask about whether there's any kidney failure in the family, whether there's anyone who has blood or protein in the urine, but you should also ask about the non-kidney manifestations, particularly those that have early onset diabetes, early onset hypertension, early onset CKD, or CKD that is unaccounted for with no obvious cause, patients with family members with epilepsy, cognitive impairment, hearing loss, or any other major illnesses, because these would suggest that it could be one of the syndromic genes that is running in the family. The other thing is that many of these family members may be asymptomatic. So the mother could be having proteinuria, but was just never been detected before. Or there may be some maternal uncle that was also having proteinuria or hematuria but never detected before. So if you can have the whole family, especially including the extended family, to come and see you in the clinic, and you check the urine of every single one of them together with your blood pressure, you probably will be able to phenotype the family a lot better and decide whether this is likely to be a genetic condition or not. Okay, so just to give you an example here, so I have this patient that presents at 16 years old with end-stage kidney disease, biopsy found FSGS. She also has hearing loss. Her sister has proteinuria, right? And she has a father who has FSGS in his 20s. So when you first look at this, you would think, okay, this is likely to be autosomal dominant FSGS or something inherited from the father. But actually, if we take a more detailed history, you would have realized that, hey, the mother actually has a sister that has epilepsy at early age with hearing loss. And the maternal grandmother has early onset diabetes and hearing loss. And the maternal grand aunt also had early onset diabetes. So with this, we then decide, hey, maybe the problem could also be from the maternal side, because this could be a mitochondrial condition. And really, when we check the genes of this patient, we really found that this patient actually has MELAS, right, which is a mitochondrial disease that is maternally inherited. Okay, so a family history carefully taken is important. Okay, now the kidney histology. So in general, the genes and the histology do not correlate, right? So we can have podosine genes resulting in biopsy of different types here, right? So you can see here, patients with podosine genes can have different biopsies here. Patients with nephrin can have different biopsy. And put it another way, right? The histology of your patients will not be able to help you predict which genes are involved. Okay, so histology has limited usage in terms of predicting which genes are involved. However, histology may still be helpful for us to detect the degree of sclerosis, right? However, there are some exceptions. Okay, so if your patient has diffuse mesangial sclerosis, then we wonder whether these genes are more likely, because these genes are more associated with diffuse mesangial sclerosis. Or if you are able to do an electron microscopy for your patients and you found glomerular basement membrane abnormalities, then you really should consider the collagen 4 genes or MYH9 or fibronectin 1. The other thing is, if we do be able to do electron microscopy, we do want to see whether there are dysmorphic mitochondria, because that would then suggest the presence of a coenzyme Q10 abnormality. Okay. Maybe I'll skip this. Okay, so next I'll go on to ethnicity. So I just like to bring out a few points, 2 points. Podosine appears to be rare, more rare in Asians. So you can see here in the Caucasian population, podocin genes account for 10 to 30% of sporadic cases and 40 to 50% of familiar cases. Whereas in Asians, the frequency is a lot lower. The other thing that it seems to be coming up, not entirely proven yet, but there may be some suggestion, is that the coenzyme Q8B gene seems to be more common amongst the Chinese. I'm not sure whether it's common among the Vietnamese. In the Africans, the APOL1 risk alleles are considered important, right? Okay, so ethnicity does have some implications. Okay, so predictors of genetic diagnosis here. So these are the important predictors of genetic diagnosis, right? So a positive family history, young age of onset, extrarenal manifestation, rapid CKD progression, early onset steroid resistance, persistent proteinuria without edema, hypoalbuminemia, and a history of persistent proteinuria prior to nephrotic syndrome. And so if your patient has proteinuria earlier on, even before nephrotic syndrome, this is actually a telltale sign that this could be a genetic cause. Okay. But of course, you do need to do some workup to see whether there is some secondary causes or not. Okay, so as we can see here in the DRAGON study, we did find that if the patient has proteinuria without nephrotic syndrome at first manifestation, they are more likely to have a genetic etiology compared to those who present with nephrotic syndrome, right? So it's 33% versus 10%. But if these patients have a second risk factor like a positive family history or extrarenal manifestation, then the risk is even higher. It is 60 to 100%. And this is fairly consistent with a similar study that was done in Mayo Clinic, where with just proteinuria without hypoalbuminemia or edema, then the patient is more likely to have a genetic cause. The other thing is the genetic cases are more likely to decline rapidly over time to end-stage kidney disease. So a rapid EGFR decline with no obvious cause will also make you wonder this is a genetic cause or not. Now, how can genetic testing help your patient? So I think the most obvious implication would be the treatment strategy. So if your patient is diagnosed to have a genetic cause, then the first thing would be to optimize the use of ACE inhibitors and ARB. Next would be how we should change the immunosuppression. So in general, we should stop the steroids if you find that the patient is genetic, because generally genetic cases do not respond to steroids. There may be some steroid responsive genetic cases in the literature, but I think many of these genetic variants described back then may actually not be pathogenic if you apply the new ACMG criteria. Would you stop calcineurin inhibitors? Well, the genetic cases can respond to calcineurin inhibitors, although these responses are usually partial and not long-lasting, and that's because of its effect of calcineurin inhibitors on podocytes. So the calcineurin inhibitor actually can act directly on the podocytes and stabilize the actin cytoskeleton and improve the proteinuria, right? So instead of working through the immune cells, it also acts directly on podocytes. So there are many benefits of doing genetic testing in renal patients that I will not cover here. I think one of the key controversies is whether genetic testing should be done before biopsy. So there is accessibility. So if you have readily accessible genetic tests and the expertise, and there's fast turnaround time, you should consider doing genetic testing before biopsy. But you should still consider doing biopsy if the genetic testing does not yield any result. You want to know the degree of sclerosis, or if there's a rapid clinical cause, meaning that the patient seems to be behaving like a nephritic case, or there is a rapid kidney impairment, then that's when you should still consider a biopsy. Okay. Now, if your patient has features of Alport syndrome, for example, definite clinical features of the genetic condition, you really can diagnose Alport syndrome clinically without the need for genetic testing. Do you still need to do genetic testing then? I think genetic testing will still have a role if the parents intend to have more children. If you want to identify the pre-symptomatic family members, meaning you have family members who may have the disease, may have the gene variant, but it has not manifested the disease yet, it is important to identify them so that you can start treatment early, and that can actually affect the outcome of the family members. Genetic testing can also guide the surveillance and treatment strategies, because if the patient is known to have a certain mutation or genetic variant, you would then do screening more effectively lah. So for example, you will look for the eye abnormalities more carefully, or look for the ear abnormalities more carefully if you know what genetic condition the child has. And eventually, if the child requires a kidney transplant, then you do want to know whether the living donor carries the genetic variant, because if so, then the living donor should not donate. Okay. Now just go through some of the important implications. So first will be the gene panel. So whether you sequence one gene only, so you just take one orange, or you do a panel of genes where you do many genes at one time. In general, the first line would be a gene panel, right? So if a patient has a genetic nephrotic syndrome, in general, we prefer a gene panel, and you really can select the genetic test at this website, and then you would look at things like the genes, the cause, the test characteristics, particularly the copy number variation, the mean sequencing depth, as well as the intron-exon boundaries. Yep, and then okay, so copy number variation is very important, because if you don't choose the test that look at copy number variation, you will be missing a certain percentage of your patients. Then you do want to see how the lab actually curate their variants. That means how they determine whether a genetic variant is pathogenic, because that is important. And really, there is a wide variation across different labs in terms of the variant curation. Okay. Now, so if an initial targeted gene sequencing is negative, it doesn't mean that your patient does not have a genetic variant. You should still look for copy number variation, and if your genetic test does not cover that, then you would want to make sure that has been checked. Then you may want to go on to a whole exome sequencing or whole genome sequencing, so that you can look at the novel genes or the atypical genes. And then if that is still not done or cannot be done or negative, then to consider repeat the analysis of the sequencing data every few years. Now there may be okay, so genetic counseling is something that you really need to make sure that you do properly. So before sending the patient for genetic testing, you do want to make sure that we have discussed all these things to the patient, okay, including what are the likelihood of a positive finding or a negative finding, and what a negative finding would imply. Particularly if there's variant or unknown significance, what does it mean? And also incidental finding, meaning that I'm sending for a genetic test to look for causes of my kidney condition. But sometimes because of the genetic test, I happen to find that the patient actually have a variant that can cause Alzheimer's when he is older. Or in girls, you realize that the patient actually has a genetic variant in one of the breast cancer genes, like the BRCA1 gene. Then what do? Right? It actually can have implications, and these should be discussed at the genetic counseling before the test. Then there should be post-test counseling, where we actually discuss the results of the test and how you interpret them, the implications of the test, the management options, alternative treatment, and some of the other things. Okay. So with this, I'd like to summarize by saying that collagen 4 genes are very important causes. A combination of risk factors can help determine the likelihood of genetic diagnosis. The more risk factors a patient have, the higher the likelihood of genetic diagnosis. Look for coenzyme Q10 deficiency, because in those patients, supplementation of coenzyme Q10 is effective. And actually, a clinician can alter the pathogenicity classification of a variant depending on the phenotype of the patient, as well as the family segregation. Okay, so with this, I'd like to end my talk, and maybe I will also end the recording here first. Just give me a moment.